And we are recording with the two kings of misinformation, which I say that obviously jokingly, as you are both incredibly credentialed and always back things up with sources and citations and are both walking encyclopedias. Neither of you are new to the podcast, but this episode with the both of you is with Dr. Peter McCullough, who has been on here who knows how many times and instrumental, if not key, in getting me permanently banned from YouTube, which I still love. And of course, my buddy George Webb, investigative journalist. But I wanted to put this together, and it's kind of like an event horizon. I don't actually know what's about to happen, but I know that George is a fan of Dr. McCullough, and I don't know, I'm kind of like a mad scientist. I want to see what happens when I mix two beakers and just let it go. So, George, I'm going to actually let you take point on this one.
Well, great. Thanks, Tommy. It's a great honor to interview you, Dr. McCullough. And, you've done a lot of really great groundbreaking work on myocarditis. I think that's the first thing that everybody looks to as one of these adverse effects. And I know you do a lot of court. You've been involved in court in terms of getting drugs approved, and you know how to present scientific technical information to a lay audience. Can you give everybody kind of an update of the latest and greatest in research you've been doing into myocarditis?
The COVID-19 vaccines, the messenger RNA vaccines, Pfizer, Moderna, are loaded on lipid nanoparticles, and then Johnson & Johnson, AstraZeneca outside the United States, the adenoviral DNA vaccines, again, also on lipid nanoparticles. They have all been shown in the peer-reviewed literature to cause myocarditis or heart inflammation. Now, our CDC and FDA officially recognized this in June of 2021. And heart inflammation is serious. Myocarditis is a serious diagnosis. Even in the initial tranche of Americans that were afflicted by vaccine-induced myocarditis, well over 90% were hospitalized. And that's been shown now in multiple series. So anything that takes a young person and forces them to be hospitalized is serious by its nature. What we've learned is that this heart inflammation occurs because the vaccines install the genetic code for the spike protein. The spike protein is the bud on the surface of the virus. It's the part of the virus that was engineered through research from the University of North Carolina at Chapel Hill and the National Institutes of Health. This is well documented in papers published in Nature Proceedings and the Proceedings of the National Academy of Sciences by first author Menachery and senior author Barrick in 2015, where they engineered the spike protein to invade human cells. This is well documented. Now, the work was done in the biosecurity lab annex level 4 in Wuhan, China, since the research couldn't be done in the United States. But the spike protein that people are enjoying in their bodies right now is a product of U.S. research. Well, these vaccines all contain the genetic code for the spike protein, and then they install it into cells in the body. And when it's installed into heart muscle cells, and the cells that look like it, they take it up, is called the pericytes. The spike protein instantly damages those cells, causes micro blood clotting, and then it incites inflammation, causes white blood cells and other factors to come into the heart. And then it manifests as chest pain, difficulty breathing. It can manifest just as immediate sudden death, as we've seen in scores of athletes. And then it sets in a process of scarring the heart. And this scarring of the heart is seen by late gadolinium enhancement by cardiac MRI. And pivotal papers by Jenna Schauer in the journal Pediatrics has shown that these scars are substantial. They are very large. Let me give you an example. Of the left ventricle, anything more than 15% is considered so large that an implantable cardiac defibrillator would be put in an adult if it's scarring from a heart attack, for instance. In this case, there's scar reported at 20%, 25%, 30% scar, massive amounts of scar damage in the heart from myocarditis. And in fact, now 200 papers in the peer-reviewed literature with fatal cases published by Gill, by Choi, and Verma. Verma was published in the New England Journal of Medicine. So this has been on the minds of American doctors that people taking the COVID-19 vaccines could suffer heart damage. We know that 90% of cases are men, 10% women. So there's something to do with probably male androgen receptors, or the interface between various aspects of male physiology. And we know the peak age is age 18 to 24, as published in a paper by Scharf and colleagues from Kaiser Permanente Northwest. But I think the bombshell paper was just published from Bangkok, Thailand. And it was a study called a prospective cohort study that the FDA wanted Pfizer and Moderna to do. So when the FDA issued the biological licensing agreements for Pfizer and Moderna, they said, you must do a prospective cohort study and tell us who is really getting myocarditis. Prospective cohort means you check the patient out before they take the shot, and then you check them out again after they take the shot to see who really gets the problem. It's a thorough study. And in Bangkok, Thailand, they used state-of-the-art equipment we would have used in the United States, EKG. They used the Roche cardiac troponin blood test, state-of-the-art echocardiography. And then in those who had suspected myocarditis, they did cardiac MRI, the best quality. And we were shocked to find out that about 2.5% of kids age 13 to 18 after shot number 2 develop myocarditis by a multidimensional definition. A large fraction of them were asymptomatic, called subclinical. 2 kids were hospitalized. That's how sick they were. But the point is 2.5%, that's about 25,000 cases per million. That's much larger than what the CDC originally projected as 62 cases per million. And this large fraction that's subclinical now has people really worried, meaning the kids and young people will take the shots. They won't feel heart damage occurring. And then the first manifestation could later on be sudden death, either while playing sports or commonly while sleeping.
Or in special ops training, as we're seeing more and more. And George, I know you're about to say something. If I could real quick, Dr. McCullough, could you just, when you talked about a product of U.S. investment and research in Wuhan, China, you said that was 2015?
Yeah, the pivotal papers published by Menachery, first author, and Barrick, were published in Nature Communication and Proceedings of the National Academy of Science in 2015. The work had been done from 2012 to 2015. All of this is summarized in Peter Bregan's book, COVID-19 and the Global Predators. He outlines 36 pandemic preparedness planning events, including these papers. And of these 36 events, 25 of them generated documents like these, and 6 of them were filmed. You can actually watch them. So the virus was clearly being engineered in a U.S. operation. Now, they were working on the threat, and in the same papers, they were working on the answer, which was going to be a vaccine or monoclonal antibodies. And they have similar programs for smallpox, monkeypox, and anthrax. But I want people to understand this was U.S. research that went bad. The research happened to be done in the Wuhan Institute of Virology. Now that Wuhan Institute, excuse me, is important for Georgetown and the audience to understand. There's a connection there because a billionaire CEO, Stéphane Bainzel, who was the CEO of bioMérieux, that's the in vitro diagnostics company from France, they built the biosecurity annex for the Chinese. And Bainzel left bioMérieux in 2011 to join Moderna as the CEO. Then he starts working with the Chinese on developing the vaccine, and they draw patents between Moderna and the U.S. National Institutes of Health.
George, this is your wheelhouse.
Yeah. Well, so Stephen Bancel would be the person who would most benefit, right, at Moderna and DARPA by a worldwide breakout for something that was being studied at the Wuhan lab, because they did build it. So I absolutely agree with that. I'd just take a little turn down the medical lane with the inflammation cascade that you described. You said the periocytes, which you're saying that in the outer part of the cell, where the ribosomes are to supposedly make the vaccine, right, from the mRNA messaging. Are you saying also that some of the messengers are getting to the actual nucleus and making a genetic change, like a CRISPRing themselves into the DNA? Is that what you're saying, or no?
Well, first, the bulk of the spike protein that's produced after the vaccines is doing exactly what the vaccines are designed to do. They install the genetic code, and that genetic code is read in the cytosol by ribosomes. And the genetic code is not disclosed, by the way, to the public, but it's theorized that it's humanized because of the ribosomes have to read humanized segments of code in order to produce the spike protein. If it was purely foreign code, the human ribosome wouldn't read it. It would. There's what's called stop codons that would stop it, and it would be a done deal. But the fact that it reads it and produces a full spike protein means it's been humanized. And in a recent paper by Nunez and colleagues from Florida International, there's striking homology between parts of the spike protein and human proteins, which is really amazing. Again, suggesting this is highly engineered. I mean, 20 or so three-dimensional segments of homology. So of interest, one of the areas that's homologous is an olfactory receptor. So it's no wonder people have prolonged loss of sense and taste and smell. There must be an autoimmune attack after large exposures of the spike protein up in the nasopharynx, as an example. Another area, multiple heart proteins have what's called molecular mimicry, meaning the body, as it's attacking the spike protein, is actually attacking normal proteins in the heart. Now, the news was broken by first author Avolio and colleagues in a preclinical model showing it was the pericyte that really seemed to be damaged by the spike protein. Pericytes are cells that support the blood vessel network in the heart. They probably more avidly take up some of the messenger RNA. We know cardiomyocytes are different. They're in the business of calcium and phosphorus handling and contracting. They're probably not going to take up as much genetic material as a support cell will or satellite cell. Of interest, even though a large mass of the heart is actually the contractile cells, the cardiomyocytes, the greater number of cells are the support number cells. And that was shown in a paper by Stein and colleagues in Circulation years ago. A lot of people don't know that. So in terms of you count up nuclei within a cardiac muscle, 70% is not myocardial cells. They're stromal cells, satellite cells, pericytes. There is a trafficking of immune cells into the heart. But your question gets to, does it reverse transcribe? This was something that was on the CDC website for a long time. It's recently been taken down, by the way, from the CDC website. The CDC said for the longest time, don't worry, these vaccines won't change the human genome. They won't get into your DNA. I can tell you, many were skeptical of this because data from Rotkin and colleagues from Stanford using human lymph node biopsies showed the messenger RNA from Pfizer and Moderna, they were stuck in lymph nodes for months. This stuff doesn't degrade because it's made with synthetic nucleoside analog caps. It's not natural messenger RNA, which is degraded after one use. Normal messenger RNA is degraded with an RNase after one use. Pfizer and Moderna are stuck in the body for a long time because of these indestructible caps by design. And so because they're so long lasting, it gives an opportunity for natural enzymes to line up other nucleotides in the cytosol and make a mirror copy of Pfizer or Moderna. And so this came to fore about 6 months ago in a paper from Malmo, Sweden. First author is Marcus Alden. The senior author and the one responsible for the research is a young woman, Yang D. Marinis. And this is a solid paper. It's not been questioned by any authority. And what they did is they took Pfizer vaccine in physiologic concentrations and introduced it into a human hepatoma cell line, a liver cancer cell line, where the liver cells are avidly taking up fluids and other micronutrients from the extracellular space. These cells readily took up Pfizer into the cytosol, and within 6 hours, the cells had reverse transcribed the code for Pfizer into the nucleus, and it was clear. Now, they relied upon a center part of the code, a 444 base pair, what's called amplicon. And that's a region that they knew could reliably report if it was into the cytochrome, the chromatin. And what experts think is, since they picked a middle segment, almost certainly the entire segment is transcribed, because the reverse transcriptase was discovered and found to be line 1. So putting this all together, until we learn more, yes, Pfizer and almost certainly Moderna do reverse transcribe. They do alter the human genome. We ought to be conservative and assume we do. And now the next questions are ones that are very important. How many cells? There must be a mosaic of cells with each shot. It can't be every cell, but how many cells and in what organs? And then once the genetic code is there, what happens? Does the human body edit it out and find it as foreign? Does it start producing spike protein in a low level constitutive basis? Is it inducible in the setting of a stress? Is there a big surge of spike protein production? If spike protein is produced, does the cell immediately die? Or is it, if it's expressed on the cell surface, does the body attack it? Recently on my podcast, I had Dr. Paginus Polycretus from Italy. His theory is, in his papers, he thinks every time the spike protein is produced on the cell surface, the cell is killed. So the body will cleanse itself of the spike protein, if you will. No one really knows right now, but all of this is not adding up to anything good for people who have taken the vaccines. All of these uncertainties now are making people even more hesitant to take a vaccine. And they should be, because there's no assurances that these are safe short-term, and definitely no assurances long-term.
Tommy, do you want to say something before
No,
I ask my next question?
No. Go for it. Go for it.
Because I want to go right to filtering and liver and pancreas damage. If let's say the spike protein is kind of causing cell death, because we've seen this high uptake with people with insulin production problems, with type 1 diabetes, high mortality, and type 2 diabetes, high mortality. Is it possible that somehow the root cells in the pancreas, let's say, islets of Langerhans, Tommy's favorite thing from med school, could they be possibly being damaged as the liver and the pancreas are performing their blood leveling functions?
It's possible, but we don't see a large number of reports for acute pancreatitis, for instance. There's been a few reports on hepatitis after vaccination. They've been all different types. They've been autoimmune hepatitis, fatal reactivation hepatitis C, hepatitis, cholestatic jaundice. But these are pretty sparse. The big ticket items we're seeing is where the lipid nanoparticles, they go into the brain. So, major intracranial hemorrhages. In a paper by Burhild and colleagues from 3 Nordic countries, it was stunning. This was published in JAMA. 7,750 neurologically devastating blood clots or strokes in people who have taken the vaccine within 28 days. That was Pfizer, Moderna, or AstraZeneca. And they carefully ruled out anybody who had the infection. I mean, these are stunning large numbers. The myocarditis numbers are astronomical. In our US CDC VAERS system, they're logging thousands and thousands of cases. And if the Manoogian paper is right, there are many tens of thousands of myocarditis victims out there right now. We know there are blood clotting disorders, lots of them. And then finally, bone marrow disorders. One's called vaccine-induced thrombocytopenic purpura. But we haven't seen much in the line of pancreas and liver. And it may just be a blood flow issue and where the lipid nanoparticles go.
Well, let's move to bone marrow, as you mentioned, and let's move to clotting then. Do you see an uptick in Van Willebrand's then with the stimulating factor 8 to cause these microclots, or is it making it all the way to the bone marrow and getting to stem cells, getting to pluripotent stem cells to do that?
No, first off, for clotting, the spike protein itself is directly thrombogenic and uniquely thrombogenic. It seems to be able to tip off the coagulation cascade without the need of platelets. We've never seen this before. It seems to activate the clotting cascade and form fibrin. And then because of the spike protein unique interaction with the fibrin, it produces what's called D-dimer. or a fibrin degradation product. And uniquely, D-dimer seems to be a signal, both in the respiratory infection and with the vaccines, that clotting is going on, a high D-dimer. And so it's a very important clue. Also, the clots take on a amyloid configuration. The spike protein must fold and is considered amyloidogenic, and more spike protein accumulates, and it becomes almost like rubber. And what you're seeing in reports now is embalmers pulling out large rubbery clots, and we can see them in my practice on ultrasound. These clots don't seem to go away with conventional blood thinners, which is very disturbing. There have been fatal cases of pulmonary embolism described, and again, in the respiratory infection with the vaccines, and then with both. Remember, many people who have taken the vaccines have already had COVID, the respiratory illness. So my practice is exploding with people with blood clots right now, and all different permutations: respiratory infection, vaccine, a couple of vaccines, then the respiratory infection. It's endless. And so anybody who's taken the vaccine, anybody who's had the respiratory infection, they have some provocative life event, like an ankle sprain and have to wear a immobilization boot, or a hip surgery. I recently had a patient who had a complicated prostate surgery and probably blood clots in the prostatic and pelvic bed. He just presented with showers of pulmonary emboli, and he never took the vaccine. It was just he had prior COVID as an exposure.
Jeez.
Do you think that could be because there's some factor in the platelet or in the clotting cascade, like a platelet attracting factor, PAF, or is it a cytokine driven, chemokine driven thing through maybe a NF kappa beta type of thing?
It's complicated, but there is a particular syndrome that's been more frequently described with the adenoviral vaccines, AstraZeneca and Johnson & Johnson. It's called VITT, vaccine-induced thrombocytopenic purpura. There must be some homology with platelet factor 4, because you nailed it. The body forms antibodies to platelet factor 4 that pins platelets against the blood vessel wall, damages the blood vessels, and causes both clotting and bleeding at the same time. So the classic presentation would be a young woman takes Johnson & Johnson, presents confused, and has an intracranial blood clot, but at the same time is bleeding from the gums and mouth. That would be a classic VITT, presenting with a low platelet count. These are very difficult cases to manage. And again, it's so advanced. It's so clear the vaccine caused it. It has a new name. It's COVID-19 vaccine-induced blood disorder.
I just wanted to talk a little bit about maybe some monocytes or some early precursors to the final white blood cell or red blood cell that could be coached or being, coached in a different direction down the stem cell chain of development, possibly.
It's interesting you mentioned that. Dr. Bruce Patterson has shown, he's the CEO of Incel DX, that the phagocytic system, the macrophages and monocytes, are trying to handle the spike protein, but they can't clear it out very readily. And so the natural infection, the S1 segment, is actually found stuck inside CD16 monocytes for up to 15 months after the infection. With the vaccines, both the S1 and S2 segment are seen within the same monocytes, at least for several months after vaccination. That's as long as he's looked, but it could be even longer than that. So spike protein becomes part of the cellular environment in the human body. And the concern would be, in Patterson's first study, is from sick people in the hospital where they've had weeks and weeks of exposure to the virus. The hope would be with early treatment, we've really had a limited exposure to the spike protein and kept all this pathobiology down to a bare minimum. With the vaccines now, we're just installing a genetic code, which we know in a paper by Vertig that the genetic code is circulating in the bloodstream for 2 weeks. And we know in a paper from Harvard by Elena Ogata that the spike protein is circulatory in the body for about a month. This is a long exposure, way longer than what someone would get with a natural infection.
Okay, that's an excellent answer. the one question I've always had is about this long COVID, and is that scarring around the lungs, and it's causing an ARDS type of situation where there just isn't the lung capacity that was before, or is it causing sort of like an arteriosclerosis with not enough blood and not a blood oxygen transfer from the lungs to the heart? What's your long COVID kind of theory right now?
Long COVID are the persistence of symptoms, which can be an entire array. We try to divide them into different syndromes. It goes back to the original research done by Ralph Barrick at the University of North Carolina at Chapel Hill. He published a paper in 1992, 30 years ago, demonstrating that coronavirus infections induced in animals, the virus can be in the body for months afterwards. And actually, in those papers, he showed coronaviruses that he's working with could cause myocarditis or heart damage in animals. So Barrick was in the coronavirus heart damage research 30 years ago. 30 years ago. Now, what we know, fast forward from an autopsy study done by the National Institutes of Health, first author is Chertow and colleagues, showed that in those who died with COVID, the SARS-CoV-2 virus is alive and replicating in the body at a low level for many months after the infection, many months. So this is a disturbing revelation that when someone gets COVID and they feel better in a couple of days, the virus could be in their body for many months. And as you pointed out, some with severe disease develop what's called an organizing pneumonia. And that's the pattern we see on CT. It's not an active bacterial pneumonia. It's considered a sterile pneumonia, but it looks for all the world like a process that we call an organizing pneumonia. And it results in shortness of breath, fatigue, chest discomfort. I have patients in my practice with this. I use a variety of medicinal approaches. And then a recent randomized trial from Israel has brought some hope, where the first time they showed that a regular application of hyperbaric oxygen may start to improve things in the lungs. But you can see how in this interview, I'm quoting the literature and I'm having people be able to find the papers. Note in interviews with our public health officials or in the liberal media where they bring on these media doctors, they're never able to quote a single paper. I think the audience ought to should readily discern who is an expert and who isn't.
Well said.
Well, I know Tommy is a maven for links. So every time you quote a paper, we'll do a lookup and we'll get the links in there. I know you've quoted about 30, so Tommy's got his work cut out.
I got a little bit of work to do after this.
But that's great. I love it when you go to the literature, because this is what health professionals all over the world need, because we get so much mudslinging and so much just ad hominem attacks on scientists who are just trying to help, who could make a lot more money in private practice, 10 times more than spending so much time sharing their knowledge and information with us. So a big, huge thank you to you, Dr. McCullough, for that. And then I can go right back in after I say my thank yous, right back into some more questions. I'm interested in these cascades. I'm interested, is there an endocrinal or a chemokindal disturbance then, where the body's balance is thrown off, angiotensin or their insulin levels, et cetera? Do you see any kind of long-term hormonal imbalances being caused by COVID?
In our first seminal paper published in the American Journal of Medicine, I'm the first author, August of 2020, we highlight the importance in the acute infection of a particular cytokine. We've never seen an infection that leads with the inflammatory cytokine interleukin-6. And it's a very strong signal. We actually published a case report from our center of a woman who suffers a cardiac arrest in the ICU, and she has a prolonged QT interval on the EKG. And in the middle of a red hot COVID infection, she suffers a cardiac arrest. And the lead cytokine was interleukin-6, which is known to prolong the QT interval. Of interest, she didn't receive any hydroxychloroquine. And at the time, people were pointing at hydroxychloroquine as a smoking gun, but it was simply COVID and interleukin-6. But why this is of interest is that the cell in the body that produces the most interleukin-6 is the adipocyte, fat cells. And it explains, I think, why those who are obese, who have such a difficult time with COVID-19, they get the viral infection, and then they get a raging cytokine storm led by interleukin-6. So that's what we know on the acute side. Now, more chronically, you're asking about endocrinopathies or disturbances. One of the things we've seen after COVID, but far more commonly after the vaccines, are disturbances in blood pressure and heart rate. And there's a syndrome called POTS, or postural orthostatic tachycardia syndrome, POTS. And what we know now is the lipid nanoparticles are distributed to the adrenal glands, to the sympathetic chain in the neck, as well as in the brain. And the production of the spike protein in some way must incite the generation of epinephrine, norepinephrine, and dopamine. Those are the three circulating catecholamines. And it's been well described in the journal Hypertension. It's been described severe elevations in high blood pressure in people who take the vaccines, particularly those who have baseline high blood pressure. So much so there can be intracranial hemorrhage. And some of you know the story of Miss Quinlan, who was on Fox News around the same time I was, about a year ago, more than a year ago, where she took Moderna on the second shot and then had a very, very powerful rise in blood pressure, suffered an intracranial hemorrhage, and needed a craniotomy and evacuation. She's been neurologically devastated since that time. We've had patients in our circles with aortic dissection, intracranial hemorrhage or stroke. I've already mentioned the Burr-Hill data. So this rise in blood pressure and the lability of blood pressure is significant. There's some type of dysregulation between the sympathetic and parasympathetic nervous system. Patients will tell you they don't feel right. The heart rate can reset 20 to 40 points higher for a long time. But my experience is over time, all of these tend to slowly get better. They may need some medicinal help, but they do get better probably as the body clears out spike protein. Now, there are sporadic cases of thyroiditis. There are no cases that I'm aware of adrenal insufficiency or other forms of endocrinopathies, but I wanted to point out those salient features of this kind of sympathetic nervous system abnormality we're seeing.
Are you seeing, you mentioned the perturbation of the sympathetic and parasympathetic. Are you seeing vasovagal syncopes where people fainting and so forth and that sort of thing?
Yeah, more so just symptomatic tachycardia and hypertension, but there are cases of vasodepressor syncope. There's just a dysregulation between the balance of those two systems. And there are no randomized trials, there's been plenty of reports. In young people, I tend to actually use a modulating drug, a beta blocker that has intrinsic sympathomimetic capabilities called Nadolol. I'm using that now relatively frequently with good success.
Tommy, I'm going to let you ask a couple of background questions because I feel like I've been peppering.
No,
but I'm interested in the committees that you've been on. When we're trying to approve drugs, like you just mentioned, you often have to give testimony from expert witnesses. And I know you've been a part of that. And this is something, as many media appearances as you've made, I don't think people know that part of your background. So could you share a little bit about that?
Sure. I've been on about 24 data safety monitoring boards. I've chaired some very highly visible data safety monitoring boards for the National Institutes of Health. Actually, for one of the divisions, BARDA. BARDA is one of the biological threat divisions. I chaired a data safety monitoring board for a nuclear threat countermeasure at one point in time. But I have a lot of experience in pharmaceuticals, devices, in vitro diagnostics. So I know the standards. And when there are excess deaths that emerge, I can tell you these programs are shut down quickly. There's no tolerance for someone dying of an experimental product. And we saw none of those safeguards in the big public program with the vaccines.
You mentioned BARDA, and of course, we all think to Rick Bright and him sitting, talking about how we could isolate a virus in, Saskatoon or wherever, some remote place. genetically sequence it, send it up to a satellite, send it to a center in Scotland, let's say, where they quickly came out with a vaccine. What, 2 days later? I think 3 days later was the case. Being inside BARDA, I think you have a unique insight. I don't know if you've ever met Rick Bright or done work with him, but that all seemed a little bit too manufactured, like a manufactured crisis to a lot of us.
It's true. I think a lot of the prosecution of the Moderna versus Pfizer lawsuit will bring all this out about what was known about the genetic code for the spike protein and when. It's pretty clear the genetic code is in the Barrick Menachery papers in 2015, so they had many years to work on the genetic code. It wasn't just discovered when COVID-19 broke out of the lab or broke out of Wuhan, China. It was well known ahead of time. These were in NIH grants, and this is U.S. innovation that was working on the spike protein. Now, you're right. NIH has BARDA, biologically active reactive division, and then the military has DARPA. And so we do work on biological threats. There's a smallpox, monkeypox program. There's been an anthrax program. And since SARS-1, there's been a SARS program. And they're working on the threat as well as working on the answer. And the answer in the Menachery papers was monoclonal antibodies. They were already working on those. Turns out those turned out to be a real winner of Operation Warp Speed. We've used those in our practice all the way through. And then vaccines. In the Menachery paper, they were just worked on a killed virus vaccine. Of course, the virus can be isolated and they can make a vaccine out of it and administer it to animals. And it didn't work too well. But later on, it was thought to just use the genetic code for the spike protein in a messenger RNA or adenoviral DNA, so-called genetic vaccines. This idea has really caught the eye of pharmaceutical companies. Recently, the companies have announced influenza vaccines that will be messenger RNA. But the problem with these is there's no containment of where the genetic code is going to go. There's no way to limit how much is taken up by cells and then for how long it's going to be produced. So unless an antigen is really benign, if one of the flu antigens, like a neuraminidase, for instance, was produced in the wrong place at the wrong time, we could really be in trouble. I don't think these messenger RNA or adenoviral DNA products, because they don't control how much antigenic stimulus have, I don't think they're going to go too far in human vaccine development just because of the safety concerns. And the other shocking revelation is on Trial Site News in work done by Sasha Latipova. She's demonstrated by using the original data that the complications of the vaccines, Pfizer and Moderna for sure, are linked to specific lots, manufacturing lots, that they're not randomly distributed. And she compares it against influenza, the conventional killed influenza vaccine. It's randomly distributed across lots, meaning someone has an adverse reaction to the flu shot. It's probably something wacky in their immune system, not the flu shot. But in this case, with the COVID-19 vaccines, it's limited to specific lots. So that tells you it's not a person problem. It's not a recipient problem. It's a problem with the product. And at the crony capitalism meeting in Washington a few months ago with Senator Ron Johnson, I was there as a lead witness. Johnson said, listen, he comes from a manufacturing background. When you have complications related to specific lots, or if you had cars that came off the assembly line, there was certain recall problems with specific VIN numbers, you've got a manufacturing problem. And now in data that was released from the EMA, we know what the problem is actually. It turns out that the majority of the lots are actually the RNA is degrading and it's not very little viable materials being injected. That explains why so many people get the vaccine and nothing happens, because they're not getting much viable genetic material. But in those specific lots where the complications are occurring, it's probably a proxy for good manufacturing, good product supply chain handling, keeping things super cooled, limited use of the multi-use files. Probably all of those things are what's accounting for someone getting a strong installation of the genetic code for the lethal spike protein, and then in fact dying of the vaccine. And the World Council for Health report, June 11th, 2022, chronicles in the US CDC VAERS system the UK yellow card system and the EU UDRIS system, WHO VigiSafe system, over 40,000 people worldwide have died of the vaccines within a few days. That's what reported to the safety systems. The deaths that occur months later probably don't get reported, but just the immediate death risk is extraordinary. Now, in that pharmacovigilance report by the World Council for Health, they point out with prior vaccines, just a handful of events are pulled off the market. We would never let something go on for 40,000 deaths. Never.
Dr. McCullough is kind of like IBM's Watson, where it just spits out perfect answers with perfect citations. It's the most uncanny thing in the world. It's the most impressive. I say it as a compliment. It's the most wildly impressive. Sorry for that lowbrow comment. I know you guys are having a very intellectual conversation. I'm just sitting here drooling watching you. Sorry, George.
Well, I know we're coming up to the end of our hour, and I always want to ask the remdesivir question as the last one. And if you look at the history with, I don't mean to be political, but Donald Rumsfeld with Gilead and the Tamiflu taint that we had, and then oops, anthrax. It seemed like this contrived thing again, this manufactured crisis thing again. And here remdesivir kind of gets force-fed and force-fit after failing in Ebola, after failing in malaria, I believe, failing a couple of times anyway. Gets trotted out again. It seems like they cherry picked the data to go from the 15 days to the 11 days. Bobby Kennedy talked about it in his book with Children's Health Defense Fund. Where are we on remdesivir? And is it they're putting all their marbles in this RD blocker? I can't remember what the polymerase blocker is, but it seems like they have a solution and they're just trying to find a problem that fits it.
Well, we'll pick up remdesivir as an RNA-dependent polymerase inhibitor. And what we know there is, there was this initial enthusiasm that it could be a repurposed antiviral. It went through clinical studies. We got to November of 2020. World Health Organization called a multidisciplinary meeting. All the clinical investigators, the ethicists, critical care docs, European Society of Critical Care followed, and the WHO did the biggest study. I mean, everybody wanted this to work, but it was clear that remdesivir did not work. It caused liver injury, kidney injury, and in November of 2020, the World Health Organization declared remdesivir should not be used in the hospital. Period. Should not be used to treat COVID patients because it doesn't work. And what happened since that time, instead of the US actually following the WHO and shutting it down, our Center for Medicare and Medicaid Services, they actually put a premium on a hospital admission if they would use remdesivir, probably because the financial deals were already made. So hospitals were getting a 20% bonus if they would give a 5-day infusion of remdesivir, which the WHO said didn't work, was potentially dangerous. And so, I think historians are going to write about this for quite some time. Talk about hospitals taking a risk because you've got a drug that a worldwide body has studied. Now, it turns out 2 years later, in May of 2022, the WHO Solidarity Group published in Lancet a meta-analysis of all the remdesivir studies, and it still falls in the lines of being neutral. It doesn't help patients at all, and if they do develop some kidney injury or liver injury, it can lead to their demise. But to have this be the featured approach, where we had other drugs, far more successful in the Henry Ford study was the early use of hydroxychloroquine in the hospital. Huge study, thousands of patients. Also, Dr. J.J. Rochter down in Florida, the ICON study published in Chest, very successful in using ivermectin in U.S. patients, inpatient. And instead of those opportunities being advanced and having, have them become part of clinical practice, they were quickly abandoned. We've never had large, high-quality clinical trials of therapy. In fact, the NIH did a very small study of hydroxychloroquine very late. It was neutral, didn't cause harm, neutral, and they gave up on it. There's been some inpatient, very small trials of ivermectin, like for 3 days at a low dose, and people give up on it. But in clinical practice, the clinical standards developed because patients did get better on these drugs when doctors use them for longer courses earlier in the course of disease at the right dose. So we have this interface between community standard of care, is what doctors have figured out works in COVID-19, supported by available sources of evidence where there's signals of benefit, but we don't have definitive trials on anything and acceptable safety. And then with remdesivir, we simply don't have acceptable safety, and we've got a worldwide body saying don't use it. So this week was the first class action lawsuit announced in Fresno, California. Lead attorney Watkins was there. I made a presentation on stage. Packed house in a church in Fresno, California. You could cut the tension with a knife in the air. Families are angry, and dozens of people have lost their lives. And the charges are going to be, honestly, tortious interference, assault and battery, just everything, because it wasn't just remdesivir being given and patients dying, but also the denial of ivermectin, the denial of full dose steroids, colchicine, full dose aspirin, good nutritional care, the denial of having family at the bedside. So all these rights were stripped away. There was no medication reconciliation. If someone's on a drug as an outpatient, it must and can be continued as an inpatient if the patient wants it. Shared decision-making. If someone's in the hospital and they say, well, I want ivermectin, they need to go get it for the patient. This business of telling patients, no, they can't get it, that violates shared decision-making. I pointed that out to the crowd, that these people getting their civil liberties stripped away when they became inpatients for COVID. This is going to become, I think, some really hot testimony in these cases as they move forward.
When you get the insert on a new drug as a doctor, which is usually an FDA warning or some kind of guidance on what the target population is and outcomes and so forth, you have these kind of rules of thumb, like a Kaplan-Meier curve that could show you the efficacy of the drug versus the risk. How did those normal safeguard processes get missed with remdesivir? It's just emergency authorization?
I think emergency use, pre-purchase of products. Once governments pre-purchase products, they actually don't want to know. They've already bought it, right? They've already bought the farm. It's like with the vaccines, they already bought them before we even knew if they did anything. Now these bivalent vaccines, of which there's no human data, and even in animals, it looks like they don't work. The U.S. has already bought them. They've already bought them. So I think pre-purchase is part of this really terrible problem, because once the money changes hands, the companies don't care. They're not going to produce any regulatory dossier. The emergency use authorization is very loose, and these products just start to be used.
One thinks of the emergent bio solutions when they stockpiled the smallpox vaccine and 60 million, 120 million doses, and then all of a sudden we have an attenuated smallpox, monkeypox. It almost seems like the solution, the cart shows up before the horse in some of these things. It's
true. We know with monkeypox, there was already pre-purchase of millions of doses of the Bavarian Nordic Jynneos vaccine, which is a double-stranded live attenuated vaccine. This is really worrisome. It's never been proven to prevent a single case of smallpox or monkeypox. We know that its predecessor smallpox vaccine was tried on about 250,000 military, and it was a disaster. It caused heart damage. We know that these vaccines are cardiotropic, and now we have this monkeypox scare. Instead of treating patients with a safe and effective drug called TPOXX, which should be the featured approach, there's an immediate discussion and pre-purchase of monkeypox vaccines. The vaccines are not treatment. It's almost as if people have lost their minds on vaccines. Every time we see a hospitalized case, everybody wants to comment about the vaccine. I said, the vaccine is not a treatment. There's two things that reduce hospitalization and death in SARS-CoV-2, in monkeypox, and all these other illnesses. One is early treatment, and then two is prior immunity. Prior immunity will always lessen the intensity and severity of illness. Those two things, not taking a vaccine.
And just to get the disclaimers out of the way, you recommend all the things that normal doctors would say: lose weight, reduce the sugar in your diet, exercise, get out and get sunshine, all these vitamin C, vitamin D as just a baseline, just so we can eliminate any attacks, from
No,
the
you're
trolls.
not eliminating anything. That exact sentence is what got me banned from YouTube, recommending vitamin D and turmeric.
All I can do is just point you to my peer-reviewed publications. That's as good as you get. And it does list in the McCullough protocol. It's called the OTC bundle. In acute treatment, it's zinc, vitamin C, vitamin D, quercetin, and then over-the-counter famotidine or Pepsid. And it's all supported by the best available evidence. Again, we're just looking for a signal of benefit and acceptable safety. We're not looking for large, conclusive randomized trials. 20 to 40,000 patients are needed. Those trials don't exist. They're not even planned right now. So the treatment of patients with COVID-19 was far more an exercise of clinical judgment than it was definitive data. And I think what America needed to have is experienced clinicians, doctors who are treating patients in Washington, leading the response, not bureaucrats that have never seen a patient who are going to try to do small, lousy trials. That's not going to help America. We don't need the NIH doing a 300 or 400 person trial. That's a waste of time and effort. If they're not going to do a 20 or 40,000 patient trial, they should just not do it. And we'll use our clinical judgment and move forward. So that's basically what they've delivered on the therapy side, is inconclusive trial after inconclusive trial. Thank goodness we figured out how to treat it and use the drugs in combination. We got so many Americans through the illness. And you know what? The word is out. There's been home treatment guides, telemedicine services. The vast majority of people are getting early treatment. A recent paper by Verkerk and colleagues, a survey of 18,000 patients, showed that throughout the pandemic, the only people hospitalized were those who got no early treatment, and they were either denied treatment or didn't know about it. They got hospitalized. Anybody who gets some treatment, they can help avoid the hospital and get through the illness. And we're basically down to a COVID inevitable mentality, not a COVID zero mentality. Pretty much everybody's going to get it, and we'll get through it.
Thank you very much. It's been an honor.
Thank you.
Thank you, gentlemen, for both of you. And I will put the links to Dr. McCullough's book, The Courage to Face COVID-19 and the Biopharmaceutical Complex. I will put that in the description, as well as your Twitter and as well as your website. And, of course, George's books, George's Twitter, George's website, all that good stuff. I'll put it in the description. Gentlemen, thank you so much for that. Thanks for doing my job. I just got to sit here and listen, so that was cool. And I'll wrap this one up. George, I'll text it to you as soon as it's uploaded. And Dr. McCullough, I'll see you in 2 minutes for the next episode, which is the culminating episode of this week. So, gentlemen, thank you so much. God bless. God bless America.